Hello and welcome to the 17th International HLA and Immunogenetics Workshop, or IHIWS, hosted by the Histocompatibility, Immunogenetics and Disease Profiling Lab at Stanford Blood Center. I am Marcelo Fernandez-Viña, chairperson of the 2017 workshop. Over the last 50 years, the format of the international workshop has changed with time. The first 13 workshops were truly wet working meetings with the contributors bringing their own reagents or their own results to the same venue, and in some instances performing tests, and in all of them performing joint analysis of the results obtained at the time. The most recent workshops consisted mostly of exchanges of reagents with testing being performed in individual laboratories, and with the workshop meeting convened only for analysis and discussion. More recently, aided by the advent of the Internet and with the use of synthetic molecules, the projects have focused on the central analysis of data produced locally by a number of different working groups who shared common interests. These workshops have served the HLA and immunogenetics field by assisting the technology transfer and individual development. Historically, the workshops have served as a reservoir of common knowledge in the field and provided standardization and specifications for naming and tracking features of the HLA alleles. The 17th International Histocompatibility and Immunogenetics Workshop will focus on next-generation sequencing and promises once again to bring valuable opportunities to the HLA and immunogenetics community for collaboration and learning. We intend to go to the initial formats, including central data collection, to facilitate analysis. With the 17th IHIWS, we hope to achieve a number of goals, including promoting new technology and disseminating knowledge of both informatics and technical skills, standardizing methods and accepting a common nomenclature, advancing the continued international collaboration begun in previous workshops by connecting with well-respected organizations such as the International Histocompatibility Working Group and with independent labs and research centers across the globe. Taking advantage of the higher throughput afforded by new sequencing technologies to analyze regions in HLA and KIR genes that have not been examined in detail, in hopes that a more complete knowledge of HLA and KIR gene sequences may improve our ability to assess donor and recipient compatibility in clinical transplantation and permit us to better understand how these genes play a role in disease association, introducing new methods for donor-specific HLA cross-matching, and the capability of having full-length genomic and complete exonic sequences will provide an exceptional opportunity to both define and precise mapping of serological epitopes. And finally, developing robust research and clinical informatics tools to address the scope of big data generated using genome-wide NGS genotyping methods and technologies. The data generated will be examined and curated by the Quality Control and Quality Assurance Committee. Contributors to the IHIWS may participate in any of 5 components, as well as in numerous projects that fall under several of the components: NGS of full-length HLA genes, NGS of full-length KIR genes, mapping of serologic epitopes, informatics of genomic data of hematopoietic stem cell transplantation. To date, we have greater than 90 individual principal investigators and laboratories enrolled in the workshop. Multiple proposed projects under the serologic epitopes and HLA NGS components are currently in progress. The requirements for several others are in the process of being listed in the database. Be sure to check the website and database for updates. Community members may participate in the 17th International Histocompatibility and Immunogenetics Workshop in many different ways, including contributing samples of interesting populations, types, etc., contributing data, performing testing for partner laboratories, performing analysis, proposing studies or projects of interest, becoming a project leader, becoming a sponsor and contributing reagents or funding, being part of the conversation by following us on Facebook and Twitter, visiting our website, and reading our newsletter. If you'd like to participate in any way, please begin by registering in the database. You may register as a PI, co-PI, or as an affiliate. Visit our website at ihiws.org and click on Download Participation Guides at the top of the page to get started. Once registered, the PI co-PI may add as many lab members to the group as desired. Additionally, principal investigators can propose a new project by contacting our program manager, Tammy Weintraub, via email. Visit ihiws.org/contact to suggest a project. Once a project is approved, a project leader is assigned by the workshop chair or by the appropriate component chair. We look forward to your participation and feedback as we move toward the 2017 workshop in Pacific Grove, California. If you have any questions along the way, please get in touch via our website at ihiws.org/contact. Thank you.